I'm going to stand in for Aidan, who normally introduces these, because I was the one who suggested the speaker for this Christmas seminar. It's Christmas seminar, so... There we go. Okay. My daughter will be very proud of me wearing her special headset. Okay, so for the Christmas seminar this year, we have an amazing speaker who's come all the way down just today, just for the afternoon, from Edinburgh to do this. And unfortunately, he will be heading back on the train later, so won't be able to join us for the Gastric Band performing at Dear Grace in a couple of hours from now. But he's going to warm us all up for our festivities later with a really exciting and interesting talk. So I've known Jamie for about 10 years because Edinburgh University is one of the other hotspots in the UK for synthetic biology. And so when I'm up there for meetings, I often see Jamie, who will present some really exciting and interesting work which borders into synthetic biology, but also lots of other subjects that bioengineering is familiar with, like development, organoids, organogenesis, and also mathematical and modeling inspired work. I'm really excited. I saw Jamie talk earlier this year on this topic of bioengineering with agency, and I think this is a really interesting concept that's right at the cutting edge right now. Just to say, Jamie has been at Edinburgh for about 30 years now and has really established himself as a leader. He is, for those engineers here in the audience, he's a member of the IEEE, so that's an engineering credential ticked off. He's also a member of the Royal Society of Edinburgh, and he's a member of the Royal Society of Biology, the Royal Society of Medicine, but also the Royal Society of Arts, which I think is a trifecta that not many people have. And he has that for his hidden talent, his evening job of being a very well-established and well-respected teacher of jazz dancing. Is that right? Yeah, so that's a pretty rare set. I'll challenge anyone else to beat that one. Anyway, enough of the Christmas-themed introductions. Let's hand over to Jamie now for this wonderful talk.
Thank you, Tom, for that very generous introduction. So because it's the kind of Christmas end-of-termish sort of talk, I thought that I'd not torture you with lots of spots on gels or Western blots or something, but step back a little bit and give more of a kind of perspective and overview, conversational sort of talk. It will cover various things that my lab has done and also other people's labs have been doing, put together under an overarching framework that I'll come on to partway into the talk. So, we all know what bioengineering is, I guess, but it sometimes helps to start with some kind of a definition. So I think what most of us are doing when we're wearing bioengineers' hats is, I'll just get away from the projector, is constructing a design system to fulfill some sort of function using living materials. And traditionally, it's odd to talk about synthetic biology with the word tradition, but I suppose even its modern form has 20 years. Altogether, it goes back to the late 19th century. People were first starting to do synthetic biology, but of course it looked very different back then. It was really a branch of chemistry and optimism fused together. And Leduc's, his first book, Synthetic Biology, was round about the time of the First World War. But as I say, that was a very different sort of synthetic biology. But in the modern era, I think maybe especially from Imperial and MIT, there's been an agenda to look at synthetic biology very much through the lens of people who are used to subjects like electronics. So, for example, we're used to thinking about a fairly inert chassis, which is a word that modern electronics people don't use very much. It comes from this era of valve or tube, depending on which side of the Atlantic you are, radios, where they were built literally on a metal chassis with the components standing off underneath with insulators or sticking up in the case of the valves. And catalogues of standard components. So just like electronics people can download an RS catalog or something and buy components off the shelf with known properties, so we have things like registries of standard biological components that were very much part of the agenda of doing all of this. And they're assembled into things, in the case of electronics, just electronic circuits. You can see I have rather a taste for old-fashioned electronics here. This is an audio amplifier, the kind that I like building rather than all of this newfangled transistor stuff. And genetically, we also tend to use the word circuit, even though nothing's going round anything typically, because of this strong analogy. And also, there's a great hope that all of this is predictable, so that you can run a computer model or something and you can predict from the ground level behavior upwards to what the behavior of the whole system is. And lots of you will, well, the younger people anyway, will have met synthetic biology exactly this way because of iGEM, which is very much constructed this way, and it works. People put together small devices from designing from these parts and sometimes build amazing things. So it does work very well at that scale. But I think it's fair to say that the predictions that were being made confidently about synthetic biology in the early years of this century haven't really come true in terms of the impact and the scale that we thought synthetic biology would have by now at a kind of real-world application, real-world industrial scale. Some things, there have been tremendous triumphs. I mean, one of the most famous is the synthesis of artemisinin using a synthetic system. But what's interesting, this is one example, but there are lots of other examples of real-world synthetic biology, is that actually mostly it hasn't been from selecting parts, putting them into a circuit, and everything behaves exactly the way you thought it would. It's been much more painful. So in this particular process, there's been, of course the later doesn't work, does it? There's been a kind of, well, you can follow the spiral here, where you have a design, build, test circuit system, which started in a different organism to the one that it ended up in, because it started bacterial and ended up in yeast. And there were lots of examples of doing the constructions and then finding out they kind of worked, but not as well as you thought, so you needed to change something. And it really go round and round and round in these sort of design, test, build loops until finally the system got there. And it's wonderful that it got there. But I think this is fairly typical of things when you try to do them for real at a sort of industrial scales. It isn't as easy as we all thought it was going to be 20 years ago. And one thing that this raises is the question about why did we want it to be like that in the first place? I mean, the word predictability might be one answer, but if you want to build things that we're good at building with electronics or whatever, well, build them with electronics. When we're wanting to work with life, it seems almost perverse that we chose to frame synthetic biology in a way that ignored almost all of the things that make life distinctive and almost forced it to be only the kind of engineering we can do with non-living things. And that seems sort of odd. And that's where I want to bring in concepts of agency and purpose. They're not concepts that we find, or yet find, I'd like to say. That's contentious, but that's all right. They're not concepts that we find in the Biology 101 or even the honours level biology books by and large. In consciousness type biology you do, but in cell biology, molecular biology, the kind of biology that a lot of people in this room probably do, we don't really find these in our textbooks. They're not there. What we find is kind of applied chemistry and structural biology. So I just want to explain what I mean by these words and then get back to bioengineering. And I'm going to start with purpose because it's by far the easier of the two words to define. So we can say that something behaves with purpose if it's doing something which is directed towards a goal, and it will adapt itself and pursue that goal even when you've started it from an atypical place, or you've tried to kind of stop it reaching the goal. So just by analogy, imagine that we're talking about a park, and it's got gates, and there's an ice cream vendor in the middle. And you observe that when a small child is put in front of that gate, it makes a beeline for the ice cream. And there are two hypotheses you can make about this. You can either think children migrate north, for example, or children have a goal of getting ice cream. And that it's testable because you can put the child at a different gate, and then if children migrate north, then you will get one answer. And if children want to find ice cream, then you get the modified behavior no longer going north, but going in a different direction to achieve the same goal. So if you want to try to find out what, if there is, purposive goal-seeking behavior, what you do is you change something about the system so that its normal path won't get it to its normal destination, and then find out, will it move towards or achieve its normal destination from a different place? That's one way of testing it anyway. And of course, anybody who's talking of parks, being on a picnic, knows just how goal-directed even very simple organisms can be. An organism is much more than a cell, but these are still relatively simple. But trying to keep a wasp out of your sandwiches in a picnic is really quite difficult. Things, at least in my opinion, don't have to be living to be goal-directed and purposeful. And an example, this is horribly topical now, of course, it wasn't really meant to be, but an example of purposive behavior is built into things like heat-seeking missiles, where they have a clear goal of finding the hottest thing they can. The purpose here is, originally a human purpose, which has then been engineered into the machine to do our purpose by proxy. Sometimes when we engineer that, we find that our purpose has been incompletely transferred, which will probably happen with life as well, so the purposeful behavior in the machine can sometimes show up in ways that you weren't expecting. Agency is the harder term to define, I think, but it's the ability to take action and to choose what action to take. And by choose, I'm not trying to mean that something has to have, full consciousness the way that you can choose an action to take. Simply that the agent has got choices, it's got a range of possible options, and it can settle on an appropriate option. And it's a prerequisite for behaving with purpose, because if you can't choose, then you can't actually adapt what you're doing to behave with purpose. And in the missile system, there are clear choices. There are the choices about changing the pitch of the flight, of the yaw. I've no idea whether missiles do anything about roll. And it's got some simple sensory systems. So for a simple system, it's inputs from infrared sensors, for example, and internal measurements like its current inertia. And all of those are integrated so that it makes decisions. So I'd say experimentally, it's much easier for us to work on purpose and goal-directed behavior than it is on agency, because if we found the purpose, there must be agency. Whereas agency on its own, I think, is a lot harder to define. Every time I try to, I end up in a mess. Occasionally, I drink a really strong coffee and go and visit the philosophy department, but it hasn't helped so far. It really, really hasn't helped. So, why am I talking about all this stuff in the bioengineering department? Well, I think one way that we can think about all of the different approaches that we can take to bioengineering is to consider where purpose and where agency lie. And for two extremes, on this slide, the left-hand side shows bioprinting. It's not our picture, but this is the idea that if what you're doing, what you're interested in where cells will go, here in bioprinting, you're putting the cells exactly where you want them, pretty much cell by cell. And there, all of the agency, or most of the agency, is lying with the human experimenter and the bioprinter, putting everything exactly where it needs to be, and the cells aren't really being asked to solve any problems at all in terms of the location. Whereas right at the other side, you have somebody sowing seeds somewhere, and what's in the seed is going to solve pretty much the entire problem of making the plant. And this is the inherent agency that's been evolving for 4.5 billion years or whatever, and we're only making small differences like deciding where to sow the seed. So I think that, I'm going to put more sort of intermediate parts on that spectrum now, and I'll show some of the work that we've been doing at different levels of this, and also bring in the work of others. So one way of engineering is to keep the agency to ourselves. I've already talked very briefly about bioprinting. This is a different form of biofabrication that we've been developing in Edinburgh, and I'm partly presenting because if people have a use for this, that's great. Please come, email, and we're really happy to work. So Ian Holland, who's a PhD in mechanical engineering, is the person who was doing all of this. He decided to try biology and ended up doing a wonderful hybrid. So this is a process called rifle. One of our bioengineering problems is ours as in mine, but also, whole field, is making tubes. Because our bodies are absolutely full of tubes. Blood vessels are an obvious example, and there are some others like fallopian tubes that get damaged by infections, or vas deferens that does, ureters that get damaged mechanically by infections, or by infections. There are lots of tubes that can get damaged that people want to fix, and grafting kind of works in some ways, but it has its problems. There are reasons for wanting to engineer tubes, but they're quite difficult things to bioprint. With cells at anything like physiological densities. So what Ian was doing was building this new system, which is actually extremely simple. So on the top left today, you can see that the big grey object is just an electric motor, whose axis is attached to a hollow perspex tube with a couple of drain holes in it and a big hole at the end. And all that happens is that you put in, this is being shown manually, he's automated the system now, but put in a syringe to do it manually and put a little drop of a still liquid matrix loaded with cells into the inside of the tube. The tube is spinning, and that causes rimming flow, which spreads out the drop into being a thin film on the edge of the tube. And you then let that gel, whether you add a chemical. If it's something like alginate, you can add calcium to make it gel, or if it's collagen, you just let it warm up. Or other things, you do other things too, but you get the point that you just make a thin layer of the gel, and then you do it again and again and again and get lots of layers. You can change the layer thickness with the speed of rotation or the viscosity of the gel, or both. And you can either have lots of layers the same. Here you're seeing lots of layers that have got different mixes of cells carrying 3 different dyes. It was something Ian did for Pride Week, but it makes the point that you've got really kind of precise loading of different cells into different layers, and that they stay largely put. All of these layers happen to be the same thickness, and also that the cells are at pretty much physiological density. And their survival is really good because they're not going through the shear that you get in the nozzle of a printhead. They're never in anything smaller than a big drop. And the forces are actually quite small, the centrifugal forces in this. So that's one way of really keeping the agency, at least of who goes into which layer, very much to ourselves. And that's a section of a whole tube. But of course, as well as making tubes, we are making layered sheets, because if you've got a tube, you just slit it endwise and then you've got a sheet. So it's a few people are interested in working so far on projects about skin, or theses which are where tendon meets bone, which is a layered structure that's hard to engineer, and a few other things. But if other people have applications, this equipment is very cheap and easy to make, so we'll very happily talk to you. Another example of keeping a lot of agency to ourselves is using optogenetics so that we can program behavior into cells which can be activated by light. So this is an example which is built by Hannes Bayer. It's part of a collaboration that we had with a group in Düsseldorf. And these are examples of transcription factors which are activated by light. So in the case of the blue light one, it's one of these love peptide domain things that comes from plants. And in the dark, the molecule is curled up and it blocks an interaction site. In the light, that uncurls, releases the interaction site, allowing that dark red little block to bind an activating part of a transcription factor, transcription activating domain, so that the DNA binding domain that contains the thing labeled love is now attached to the PDZ and the VP16 domain, which activates transcription. And then there's another system on the right that Hannes built, which can be triggered one way by red light and the other way by far red light. And we can, for example, this is using that system to control cell death for apoptosis. So here you have a projector which is projecting onto a lot of cells, a sort of hand shape. It's only a 4-fingered hand, but it's basically a hand shape. And you can see with the live dead staining. So the GFP is the living cells, and the cytox, which is the blue, stains dead cells. You can see that after exposing all of that, the cells will die where they've been flashed with the light. If normal cells don't die with this light, it's just a very brief exposure of actually very dim light. So there are lots of other ways we can keep agency to ourselves, and there are some real advantages in it. One is that obviously we keep close control. That's the whole point of keeping agency to ourselves. We can say exactly what happens, and we can think pretty linearly, at least compared to where I'm going, we can think pretty linearly. There are disadvantages though, and one big disadvantage is that we have to do a lot of work, and we have to do it every time, or program a machine to do it every time. Because if every new batch of cells has to be told exactly where to go or exactly illuminated to tell them to do something, we're keeping a lot of the hassle to ourselves. There's also the disadvantage that we can only do things that we fully understand. So for example, if it wouldn't make any sense if we wanted to herd sheep. In principle, you could imagine wiring up all of the muscles to a sheepdog and controlling it manually with some complicated remote control thing. But in practice, that would be, apart from being mechanically very difficult to do, we don't understand enough about sheepdog locomotion or sheepdog behavior that we could do that. What we choose to do instead is realize, oh, dogs are actually quite good at moving sheep around. They know how to do it. What we need to do is to communicate with the existing agency the dog has and use it so that we can give commands to the dog, and the dog will mostly run itself in terms of rounding the sheep up. And essentially, we're telling the dog where we want the sheep to be. Unfortunately, our ancestors realized that a long time ago, which is why we have people who are wearing wool for the winter. That's why we can do that. So it's difficult to control complicated systems. And as well, by doing all of this, we're still working quite away from what makes living things most distinctive. So at the other extreme, we can work with pre-existing agency. And an example where I've been doing this for a long time is in organoids. So I just want to introduce a particular organ, which is the kidney, before I talk about the organoids, the natural kidney. So this is a sketch of 3 stages of kidney development. The first one, which is on the left, is how the kidney starts. So it's really got 2 populations, or 3 populations of stem cells. It's got the pale blue thing, which is labeled ureteric epithelium. That is an epithelial tube, which is going to branch and branch and branch and branch and branch and make a tree-like urine collecting system, finally. It's got some green cells around it, which are the precursors of all of the excretory nephrons of the kidney. Those are the tubes that at one end have the blood filter, and they then recover things that we don't want to pass out in urine, and they connect to the urine collecting duct system. And then the purple, the magenta color is precursors of stroma, connective tissue, and bits of blood vessel and so on. So that does its thing of branching and making the black branch tree in that middle drawing, and the red things are the nephrons. And then eventually, at a much larger scale, the kidney grows and matures. This is a detail of the cortex of a developing kidney. It's just a section, and you can see that what you're seeing here is part of in blue, you're seeing part of the branching tree. It's looking, I mean, you can't see that it's part of a whole tree just because it's dipping below the plane of section, but that is all part of a contiguous tree. And you're seeing around the 2 top branches of the tree in green forming nephrons. And on the top right, you can kind of see that vaguely S-shaped object, which is a slightly older nephron and so on. That's what this looks like. Well, some years ago, about 2009, a physicist called Mathieu Ambacant came to my lab, and in one of those sort of far too late in the evening after long cell culture conversations, we were talking about wondering what cells knew. Just one of those kind of, very, neither of us did drink alcohol, but almost a pub conversation of, well, what do cells know anyway, So that was kind of brushing against what I'm now talking about in terms of agency and purpose. And what he wanted to ask, or what we sort of cooked up together, was asking these stem cells right at the beginning. They're all in an anatomical relationship with each other, and they end up producing the sort of anatomy that I've shown you. What happens if we do the equivalent of moving the child to the other gate and really change something about them? Can they still get there? So his simple thing was just to take them apart into a suspension of single cells, hurl that together as a random lump just by centrifugation, and culture the lump. So when he did that, What he got was something that, again, looked very much like the cortex of a normal developing kidney. So there were little, in this case stained green, treelets of what will become the collecting duct system, the ureteric bud. So some of them are branched, some of them are not branched yet, and shown in red are the developing nephrons. So that complete mess of randomized stem cells managed to organize itself to make these things, which we thought was fairly exciting. But when we looked, wherever we looked at the lump, it looked like this. So it was symmetrical in the sense that everywhere was boringly the same, and that's not how kidneys normally develop. Normally there's organ scale structure as well, which is why earlier I said this is what the cortex, the outer part of the kidney, looks like. So what that was sort of telling us is, well, cells do know how to do something. They've certainly managed to generate the microanatomy, even when totally messed up and randomized, but what they don't do is generate the macroanatomy. So they know some things, they don't know everything, much like us. We could push on with this a little bit. So a PhD student in the lab, Veronica, thought, well, maybe one of the reasons that the anatomy is not right at organ scale is because we've got all these little ureteric bud treelets that form rather than a single tree. In real development, we had all of this coming from a single progenitor tubule, the ureteric bud, which then branched. So what happens if we make one of the organoids that Mathieu made, which is what's happening down on the left, then take one single treelet from that and mix it with all of the stem cells except stem cells that can make the ureteric bud to make a second organoid. So now we've got a single source of tree-making material and still randomized everything else. And in that case, what you end up with is an organoid that's made around a single tree. That's more realistic, and you can see that some of these nephrons are actually attached to the tree. You can see them joining top right of that bottom picture, for example, you can. But the main thing that's wrong with that tree is there's no way out. If this were ever going to be a working kidney, which of course it isn't, everything I'm talking about is being done in a dish, but if it were, it would be a very useless one because there's no way urine could get out of it. And that was addressed by another PhD student a few years later called Chris Mills, who again went back to the embryo and asked, well, why does one part of that tree develop into something else, the ureter, the trunk of the tree, if you like, rather than developing into branches? He looked at the signaling environments around a developing kidney and found an asymmetry there, and he basically found that a region which expresses a lot of BMP4 is where the ureter develops, and everywhere in the kidney you have an expression of gremlin, which is a BMP antagonist. So he ended up doing a simple experiment of having a bead that would release BMP4. Obviously, there were control experiments releasing other things. But this experiment, where you can see the asterisk, that's where a bead was. You may, if you're near the front, you can just about see the outline of the bead around the asterisk. That was releasing BMP4 next to one of these developing Veronica-style organoids. And in that case, where the bead was, formed a ureter, expressing uroplakin, which is the orange color, which is characteristic of ureter. While far away from the bead, we had the branching and we had the nephrons and we had the formation of something like a kidney. So I mean, these are very small. These are tiny compared to real kidneys. I don't want to exaggerate what we've produced. But anatomically, they're much more like real kidneys. So this is an example of working with the inherent agency of the cells. They know how to make kidney tissue, but intervening by doing two symmetry breaking operations. One, the Geneva making a single treelet, and then the other one, this slow release bead, to break the symmetry of the system twice to get something much more kidney-like. So that's kind of natural agency plus human intervention. Using natural agency is a very ancient way of engineering biology, and I know there's at least one surgeon in the room. This goes back a couple of thousand years at least. It really got going dramatically, I think, in the development of plastic surgery, where the simple property that if you damage two tissues, if you wound, if you cut tissues and you oppose them, they will normally heal themselves together, was used in the beginning of advanced plastic surgery. For example, in World War II, the Guinea Pig Club, these were mostly RAF airmen who'd been badly injured, usually burned in aeroplane crashes. And in this picture, you could see somebody who's having his face reconstructed by taking a graft from his arm to attach to his face, so that the skin which you're seeing attached to his nose is at the moment still having all of its blood supply from the arm, which is why it's alive, but it will heal into the cut skin in the middle of the face, and eventually will be getting all of its enough blood supply from there that it can be released from the arm and resculpted to make the new nose and centre of the face. And that's something which was developed in World War II and has been advanced more and more, and is using entirely the natural agency of cells just to heal when they've been damaged. The same thing is used, of course this is still used for accidents, it's also being used, for example, for people who are making transitions between gender. So this is an example, a very similar one. The paper shows all of the stages. If you're squeamish, don't read the paper. But this is somebody, he, as I would now say, post-operationally, had skin taken from his arm, where the skin was first grown around a former to grow to the right kind of shape to become a new phallus, a penis for the person's new life as a man. So the advantages of using existing agency is that we don't have to do a load of genetic design and manipulation or anything else like that. And of course, clinically, that's really good because we don't have all the safety concerns of doing a lot of GM. Evolution has made a lot of these systems very robust. Also, we may be able to discover a few Easter eggs. So what I mean by that, Easter eggs are these things that are put by programmers in software packages. So for example, in early editions of Excel, there was a maze game, an adventure game that people could play. You typed in the right formula and it would launch an adventure game, and if you just touched escape, then it would go back to a boring spreadsheet in case your boss is walking behind you. And it's been a long game of software developers to do this kind of thing, just hidden gems. And Easter eggs are probably also in our cells, things that our cells can do that you wouldn't know. This has been explored a lot by Mike Levin and his colleagues in Tufts University in the USA, where they've been taking cells from amphibian embryos and putting them in dishes. And for example, if you take the so-called animal cap of the amphibian embryo, which is an area that's going to form neural tissue and skin, and put it on its own away from the embryo in a dish, it runs around, and they've found ways of shaping those little lumps of running cells so that if they give them disaggregated cells of the same type, they bulldoze the disaggregated cells to make the same shape, and those cells are then a new aggregate that run around and bulldoze some more. So you've got this kind of as long as you've given them the cells to work on, a self-reproducing system that has nothing to do with normal frog development. Whatever, that behavior couldn't possibly have been selected for as part of normal frog development, because these cells don't run around at all in normal frog development. It's that it's a hidden property, which if you take the cells out of context and put them somewhere else, they do something interesting. Of course, the disadvantage of working this way is that the properties you want may not exist at all. And if they do exist, whether we find them or not may be almost random. It's difficult to predict what properties things have. So another approach we could take is thinking, okay, we quite like the idea of working with agency and purpose. We don't know if we'll find it, so why don't we engineer purpose? Instead of engineering all of the details, let's engineer the purposive behavior and then let the cells use that to sort the details out. So engineering new purposes into cells is the next bit I want to talk about. And one thing that I want to say is that the point that's needed, the purpose shown by a cell may not be the purpose that emerges at the level of a whole system. So here, this is a picture, a famous movie of a neutrophil. And even the most kind of, against purpose and agency people I know still find it hard not to think of that cell as hunting those bacteria when it's been chasing them around. This cell clearly has that kind of chemotactic, I want to close in on the bacteria and phagocytose them behavior. At the level of a whole patient, the purpose is to get better from a disease. It's a slightly different thing. So the behavior at the cell level that will achieve the behavior at the high thing, it's a different kind of purpose altogether. Chasing bacteria is different from getting better and healing yourself. Do you have the means of advancing? Thank you. Thank you. Okay, so that was the point I was making. So what we did to start exploring this was to make pattern-making systems. And the pattern-making was using cells that had a simple purpose, which is that they would bind to their own kind. So we started with hex cells, which are simple mammalian cell line, and engineered the hex cells in two different ways. So each one would have a homophilic binding molecule that would bind to its own kind and would also be fluorescent. And we wanted to do that because we reckoned that it would achieve so that the purpose at the single cell level of each cell is just shuffle around and bind to something like U. But we predicted, I'll explain why in a moment, that that would generate at a multicellular level a different kind of thing, which is a pattern-making purpose. And this came from just a simple computer model of what happens if cells if cells want to bind to their own kind and can move around. So want to bind means that it's energetically favorable to do so, which is what adhesion means, really. And the computer model just predicted that what you get is relatively stable patches, because the cells would start to separate to find their own kind. But then, for example, cells in two different yellow patches here can't join up easily because cells would have to leave the yellow and get into the blue region, which is energetically unfavorable for them. So it's a relatively stable pattern. And what we can do, that would be a stable pattern if it were formed. What we can do is to start with random mixes of cells and ask, do you get pattern formation in that case? Do we get back to that? And the computer model suggests, yes, we do. We're started just then with a random mix. This isn't actually the model running live. It's a movie of the model running, but you get the point. That what the cells are doing is moving around, shuffling neighbors all the time and forming a patch pattern like that. That was the prediction. So Elise Cachat, who some of you probably know because she's a PI herself now, made these cells. They're really simple, just a tet-inducible promoter, an adhesion molecule and some fluorescence, and asked, do they make this kind of pattern? And the answer was yes, they do. So this is a dish with exactly those kind of cells. They started off random, and they made this kind of patch pattern either in 2 dimensions or also in 3 dimensions. If it's really small, then in 3 dimensions it'll sort out completely, but anything more than a few thousand cells, you end up with the pattern. And for Kian Lekifridis, who was a PhD student with me, who's now a postdoc in the States, did the same kind of thing but in reverse. So he made cells that have heterophilic contacts, so 2 different types of cells, and they want to bind to the other one, not to their own kind. So these are xenophilic rather than xenophobic. And what that does is create hyperuniformity. So it means that, for example, the orange cells are statistically more separated from each other than they would be if they were simply random. Other people have taken this to to much more impressive levels, I think. So Wendell Lim's group made a patterning system which came from a single cell type that had got a feedback system so that the receptor SYN notch there, when stimulated, would make the cells sticky and glow green, and make a ligand which would and not make that ligand. Without the SYN notch being activated, that ligand would be the CD19. The CD19 would activate the SYN notch of a neighbor, but a weird property of notch is that expressing the ligand on the same cell is inhibitory rather than activating. And that meant that the one single cell type would spontaneously break into 2 different states, the red state, which isn't sticky, and the green state that is, so you get this shuffling around, so the sticky cells are in the middle. So that was from a single type of cell, which I thought was very impressive. And Mickey Abasia's group made a Turing wave type system, again with a single cell with patterning based on Alan Turing's ideas, where you have a slowly diffusing activator of its own expression and of the expression of a rapidly diffusing inhibitor. And that creates the reaction diffusion system, which creates pattern. And both of those things have been published. And we can also we can drive morphogenesis off the pattern. So, for example, if going back to Elise's patterning system, if we let these cells make their own pattern, which they're doing by the agency that we engineered into them, and we've engineered the green cells to die if in this case tamoxifan's put into the system, then we get the pattern, we then eliminate the green cells, and we end up with a structure. Admittedly, a flat sheet with holes in isn't the most impressive example of morphogenesis, but it's a start. We'd like to do fancier things. So I think the advantages of engineering agency into cells is that we don't need to talk to the cells real time. Once we've engineered the cells, our work is kind of over. It may be easier to scale all of this because we don't have to talk to the cells every single time once we've built them. And it may be easier to build extra functions on. So I've illustrated that by having that apoptosis to make the holes engineered on top of the pattern formation. It may be easier to keep going that way. The design challenges are more complex, though. We can't think quite as linearly as we were when we had all the agency to ourselves. And particularly if we were trying to make something which isn't just a proof of principle, I think the design challenges are more complicated. So we can also work in mixed mode. An example of this, again, this is for Keir and Lekifridis. What he's done here is taken one of the cell types that you saw on the patterning system, the red one. So these are cells that stick to each other with a cadherin called CDH3. And CDH3 is a molecule which it's found in the placenta, but it's not found in many other places in the embryo. And in particular, it's not found in the early embryo. The early embryo, or embryoid bodies made out of embryonic stem cells just aggregated together on a dish, express CDH1, which is a different cadherin. It was the one that was expressed in Elise's green cells. So CDH1 cells like sticking to each other, CDH3 cells like sticking to each other. They will interact with, 3 and 1 will interact weakly, but they won't invade each other's territories. So what he did was engineer those cells to express a signaling molecule, in this case Wnt3a, and then he added the cells on to wild-type embryoid bodies made from embryonic stem cells. The reason he was doing that is that if you take embryonic stem cells and you aggregate them to make an embryoid body and let it differentiate, you get cells that represent the 3 germ layers of the embryo, the ectoderm, the mesoderm, and the endoderm. So that's stuff that makes your whole body, effectively, between them. But instead of the beautiful organization you see in a real embryo, the production of those cell types is chaotic in an embryoid body. And what Fakir wanted to do was to introduce some order to this. So what he'd done by making his cells, which will make aggregates spontaneously and will secrete the signaling molecule, is effectively make a synthetic organizer. Because if you just mixed these in with embryoid bodies, they'd spontaneously stick to the embryoid body, and they'd stick onto the side, so you're seeing them glowing red here. And because they're secreting the Wnt, they would start getting the ES cells to respond by changing their own gene expression, but this time in an ordered way. So this green gene that's coming up, which is, in this particular case, it's a GFP marker for Wnt activity, but later pictures in the paper show natural genes coming up like brachyury. These things are coming up in a gradient away from the synthetic organizer. So the organizer are cells that he's engineered to make this little blobby organizer to stick on the side of an effectively wild type, apart from its reporter, ES cell embryoid body, and to impose order on that. And that's something I think, it's an interesting way of working mixed mode, because it means that we're engineering organizers for wild type. And if what we want from our embryo or our stem cells is tissue that we might eventually use clinically, we want it to be wild type. We don't want it to have lots of difficult genetic engineering that's going to be a nightmare to get approved. So I think this is an interesting kind of approach. And as embryo models get more and more complicated, I mean, you have human embryo models now that make somites and neural tubes, so effectively spinal cord and the progenitors of ribs and vertebrae and things. Having controls in is probably a good idea for ethical as well as technical reasons. And then you can get really mixed mode. So this is an example of what I've just said, of having synthetic organizers very much of the type that I just mentioned. But this time, their synthesis of the signaling molecule isn't constitutive, it's put under optogenetic control. So again, the organizers are making the same molecule, the Wnt3a, And this is an example of two organisers stuck onto the same embryo body. That happens randomly, because remember, we're just mixing these and letting them swirl in a tube. So some of them will have no organisers, most will have one, a few will have two. This is an example that's got two. So on that left hand of the actual pictures, not the drawing, you can see there's a kind of Mickey Mouse ears shape where the two red Mickey Mouse ears, which are the synthetic organisers, and the green in between, which is just the responding cells. One of those was flashed with light to induce its production of Wnt3a, and the other one was left in the dark. And then we have the response in the responding cells only next to the synthetic organizer that was flashed with the light. So this is an example of where we've got wild-type responding cells. They've got their reporter gene in, but nothing else has been messed about with them. We've got our engineered synthetic organizers, which know how to organize themselves, but yet we've got then back to the human agency required about which ones do we choose to light. So this is a really mixed mode way of bringing agency in at different levels. The advantages, again, we can use wild type, and we can probably dodge the worst of the disadvantages of all of the individual modes if we're lucky. But the risk is that we end up having the disadvantages of each of the modes. I mean, this is you know what it's like when you're trying to build something. You always hope the brakes will go with you, and sometimes they just don't. So overall, and the sort of pattern that I wanted to give in the perspective is that we can consider there's a spectrum with respect to agency. And when we're starting projects, we can choose at the beginning, where do we want the agency to be? Do we want it to be right at the right-hand side of this, where we are going to be control freaks, and we're going to keep charge of everything, and therefore accept all of the work that we have to do? Do we want to be at the left side, where we're using completely natural behaviors, like and slightly steering them, like the natural ability of those cells to make kidney organoids with beautiful structure if we just give them a few little influences, or those swirly things are xenobots that I was telling you about from Mike Levin's work? Or do we want to be somewhere in the middle, where we're engineering new purpose into cells, and then letting them do much of the problem solving, like letting them make their patterns? We give them the purpose of behavior, then we let them do their thing and make the pattern. And we accept that detailed pattern will be different every time, statistically be the same, exactly the way that we all have fingerprints and we agree we have fingerprints, but the details are different. So if one of you is going to kill me for saying all of this, you'll get caught. Okay, having given that perspective, I shall stop now by thanking the various people who funded the work in the group, collaborators, particularly Matthias, Christian and Mustafa with all of the recent optogenetic work, and my own PhD students and postdocs. They're not all there at once, they're from various generations of them, and the university for putting up with all of this. Thank you very much for listening.
So much, Jamie. That was wonderful. Really great. Very amusing slides. So, do we have any questions for Jamie? We've got about 5-10 minutes left over now for questions. I have one urgent question myself, so I'll go first. With the park and ice cream problem, is there any way to program children to go north rather than the ice cream van? This is a very important question for me.
Okay, that's your next research project then. Not sure who'd fund it.
Yeah, me probably from my own pocket, from the savings I would make. Yes.
This is obviously being surgery, being far too low-level thinking. So the question I have is, for me, I'm very interested. We were talking about this earlier, that how do nerves know where to go, which part of the hand to go to? From what you were saying, do each cell type depend on other cell types being present? For example, does the nerve need to have some bone cells nearby or some muscle cells to know where it slots into to make that kind of agency and that purpose that you've described there?
So the general answer would be yes, it needs other cells to be present. I suspect that for a growing nerve, it won't be so much the event, the whole pathway probably needs to have information on it. So there probably needs to be a map that it reads, because a nerve in development, a nerve coming out from the spinal cord, going all the way to the fingertips, even when the limb bud's quite small, that's a very, very long distance. And the difference between going from the thumb and the little finger is a very small angular distance compared to the problem of getting out there at all. So I suspect that they're growing along pathways, just as we know full well they do in, say, the optic nerve going to the brain, that are patterning the whole route, and then it'll only be right at the very end that the final target matters and starts giving, for example, chemotactic signals. I don't know whether the bones would be needed. That's a really interesting question. I'm thinking in people who had phocomelia, so this is the drug thalidomide that was given to pregnant women in the 50s. One of the effects that it had was that the long bones of the limbs didn't grow properly, probably secondary to a blood vessel growth problem, but often the hands would grow, so you'd have extremely short limbs, but still the hands, and missing bones and things. The hands were innovated, I think, so however that worked, it didn't depend on normal bone structures along the way. Okay. I've probably just made the answer even worse by thinking about that. I'm sorry.
You give me something to think about. Thank you.
I have another question from me. Do you think there is any way, given a culture cell type or something, so for example, if we isolated cells, to kind of quantify the agency potential of a cell type? Or is that just too complicated a thing to measure? I'm considering this because there's a lot of efforts to do things like human cell atlas to understand all the expression and capabilities of different cells everywhere in the body, and it might be interesting to tie that to the capacity of cell types to have their own agency.
Yeah. So I think there will be a huge amount of what we might call housekeeping agency. So the choices that a cell can make about its perfectly ordinary metabolism, about the progress through the cell cycle, about how much to grow, about whether to go into S phase or whatever. There'll be a huge amount of that. If we had a magic agency reading machine that could look at every time a cell had got a menu of choices and was making a kind of rational decision, there'd be a huge amount just to be alive. And I suspect quite a small additional repertoire for doing fancy stuff like making a human brain rather than a loaf of bread as a yeast might. And so if we were going to quantify, we'd need somehow to quantify only the stuff that's on top of the housekeeping, or the quantity I think would be swamped by everything we have just to be living cells at all, without being able to do anything fancy. That's pure prejudice. These are my thoughts, but since I still haven't found a very good way of defining agency in a measurable way, I really don't know.
Well, I think that experiment the person did in your group, where you sort of blended up the cells, mixed them back together, and showed, micro scale patterning forming. I can imagine being able to do something along those lines for the various cells of the various tissues around the body, and getting some sort of handle on, let's say, kidney and liver are quite good at doing it, but yet, maybe thymus or somewhere would be terrible.
I think if we come out with some exact assay and at some relatively clear score about the numbers of cells and the number of cells that develop and the number of correct relations to neighbours divided by the number of wrong relations or something. I'm making this up, but yeah, I think if we sharpened the question right down, then yes, you could be quantitative, and we'd accept that there's a lot of other stuff that we're not measuring because we're only measuring this sort of purpose and agency, and I think that could be a really useful thing to do, especially with the cell atlas, because we may be able to correlate with numbers of receptors for signals being expressed, or numbers of, I don't know, things that we think are important to development.
Okay, great. Riley.
When you talk about giving cells agency, you can give sort of direction to a dish of cells, but how effective is it that they all take up that, say, instruction per se? You give something a genetic transfect, you get depending on what it is, very variable responses. Is there a point there where maybe the agency you give it isn't accepted very well at all,
Yeah,
and a
so
scale up is very difficult?
That's a really good question. So everything that I was showing, these are all clones of cells where we had, we transfected them and we then cloned them so that we knew at least they all started having exactly the construct in that we wanted to. And what we were asking of them was really simple, and they were only interacting with each other. So there, essentially everything worked every time. But the nice thing about doing the proof of concept study about something is you can always choose the lowest hanging fruit there is. And I'm very aware that doing something properly useful, like trying to wire up, a nerve to some, that's a whole lot harder. Yeah. Certainly, I mean, something that plagues the general organoid field where people try to go straight from stem cell to organoid is that usually we get loads of cells. No, I don't just mean we, I mean the whole field, get loads of cells that are the wrong thing. Kidney organoids are full of neurons that shouldn't be there, whoever makes them.
Okay, a couple more questions, and then we need to finish up.
So my question is, can these kidney organoids can be used? I mean, for example, if the kidney is damaged and we got the kidney organoids, can it be used to replace the damaged part of the kidney?
I'd really hope the answer will one day be yes. At the moment, our really big problem is that we can't get a properly arranged blood supply into those organoids. We can get them to be vascularized, but randomly. And in the kidney, the relationship between the urine-carrying system and the blood-carrying system is really important in the way that the kidney works. So we have to solve that problem. In terms of putting an organoid into a damaged kidney rather than replacing a whole kidney, there's also the problem that we'd have to find a way of plumbing the urine-collecting tree of the organoid into the main one. Actually, we found in culture and have published that spontaneously, if we put organoids next to bits of kidney, they will join up. So that's quite hopeful. I don't know if it'll happen inside somebody, but in a dish anyway.
But if they join up, will they start communicating with each other, or?
We only looked for the anatomical joining up of the two draining trees. That was solely what we looked at. It wasn't we weren't looking at function because there was no blood system or anything to be making urine. We were just asking, does this pipe join that pipe?
Thank you.
Okay, and then the last question is from the furthest away person possible in the entire room.
Thank you so much. It's been really interesting. I just wanted to return to the sheepdog analogy, actually, and the idea that you want to sort of harness their natural impulse to herd sheep. I'm a cat person, but I understand that one of the most important parts of herding is to have the dog also stay and not herd sheep when it's not appropriate. So if we subvert sort of the idea of agency and purpose to tumorous cells, for example, because a lot of the work today seems to be along the developmental side and understanding, so organogenesis. I wonder how you might adapt these concepts of giving purpose and agency to taking away purpose and agency in engineered systems to understand cancers.
I think that's I think there are two really interesting aspects of that. So taking it away to understand cancers, yes, absolutely. At the moment, people work very much at the level of individual genes. I think higher level views could be very interesting. Also in terms of agency and affecting how cells behave, there's an experiment which has always intrigued me, which was done in the 1960s, and as far as I know, no one's returned to it. There's a still extant Xenopus kidney cancer cell line. And somebody in the 60s did an experiment. If you cut off the growing limbs of tadpoles, they regenerate. This cancer cell line, if you put it into frogs, it'll just multiply, or tadpoles, it'll just keep growing as a cancer. If you put it in the blastema, the regenerating lump of cells in the limb, it forms limb, it doesn't grow into a cancer. And I wish somebody'd return. I'm not a Xenopus person. I don't have the facilities, but I wish somebody'd return to that experiment to find out, was that just a bad paper and someone made a mistake, or was it true? Because if it is true, It's telling you if you're taking a full-blown cancer line, but you put it in a weird environment where it's having to mature into something else, it's now fine. It wouldn't be fine in a kidney when it found, it would probably be cancerous again there. But I think that idea of kind of differentiation therapy, of kicking a cancer cell down a different line, and then it behaves because it doesn't even need the gene that got mutated, is a really interesting one. And it's one that's sort of been slightly forgotten about. I think from the 60s to the 80s, people talked about this a lot. And then they've gone back to the idea of just killing them. And the problem with just killing cancer cells is you're always fighting against resistance. And whereas getting them to behave and to do something else that they can do, I think is just one of those approaches. I'd like to think some people were still working on it somewhere in the world. Thank you for your question.
Okay, everyone, let's please thank Jamie once more time.
Thank you.
Jamie will probably be around just for a few short minutes, but he's making the wise choice to try and get back on the tube to King's Cross before, well, Friday night before Christmas on the tube can get very interesting later on, as Londoners will know, at this time just before Christmas, yeah. So thank you very much. It's been a pleasure to have you here, and we really enjoyed your talk. Thank you so much.